Good morning. My name is Meg Detloff, and I work for USGS PNAMP. Thanks for joining us for the first PNAMP Fish Monitoring Workgroup meeting of 2024. Whee! Let's see, some housekeeping tips before we get started. Unless you're speaking, please turn off your camera and microphone by clicking the icons on your Teams platform. If you don't see the toolbar, just try moving your mouse a little bit and hopefully it will pop up. It should be something similar to the bar that's on my screen. Just kind of depending on the way you're using Teams. So if you're using it on the browser or the desktop, they kind of tend to vary. If you have questions or feedback, please use the chat function or raise your hand and unmute yourself. If you haven't heard me say it before, we can mute you, but we cannot unmute you. We're recording the meeting and it will be available on our YouTube channel. Quinnell's going to drop the links in the chat, so she'll drop a link to the YouTube and then a couple of other stuff as we kind of go through the meeting. If you have troubles with the chat function, here's a couple of troubleshooting tips. Try leaving the meeting and joining again. Try using a different browser. Edge works better than Chrome. Try using the desktop version instead of the browser and vice versa. If you don't have access to chat, please feel free to unmute yourself and ask questions or add comments. PNAMP commits to fostering an open and welcoming environment free from harassment, so please remain respectful. I would like to acknowledge all of the Fish Monitoring Work Group leadership. The names are associated with the tasks that they have previously or currently lead. The core team, if you guys could turn your cameras on and introduce yourselves, that's great. Everybody can get an idea of what your beautiful faces look like.
I'm Nancy Leonard from Pacific State Marine Fisheries Commission.
Thanks, Nancy.
And I think there's my camera. There you go.
Yeah, there you go.
Okay. Yeah.
I'm Jen Bayer, USGS, fresh from shoveling snow. So nice to be here. Thanks, Meg.
Casey, I saw you were out there.
Casey Bleasner, Oregon Department of Fish and Wildlife, out of the LeGrand Fisheries Research Office.
Mari, you're part of our core team. You can't hide.
Oh, goodness. Okay, well, here is the hiding in my friend's attic. Hello. Hopefully the power and the internet stay on here because it's been a fun week.
Fingers crossed. Thanks, Mari. And then we also have Marika Dobos and Russell Scranton. Are you guys out there? All right. No worries. And Laura Erickson, PSMFC, she's also part of our core team. Let's see here. Thanks, core team. So this is you guys' turn. Don't worry about turning on your camera or your microphones. Just simply open up the chat and introduce yourself with name and organization. This helps acquaint you with the chat function and helps us keep track of participation. And while you guys are doing that, I'll just quickly cover today's agenda. After I wrap up, we'll kick the meeting off with updates on current tasks. Next, Ralph Lampman will give a presentation on all of the stellar work he is doing with lampreys, followed by Q&A with him. And then we'll close the meeting with just a few next steps and wrap-ups. And before we jump into task updates, I want to give a quick shout out for the Washington, British Columbia, Idaho AFS meeting. They're doing like a pretty joint session this year, so that'll be really neat. Quinnell drop a link in the chat for the homepage of the workshop. I just wanted to let you guys know that PNAMP, StreamNet, and Fisheries and Oceans Canada will be co-sponsoring a symposium on data longevity and reproducibility. A link to the symposium abstract will be in the chat too, so if you guys are interested in that symposium, I mean, feel free, if your work aligns with what we want to present there, just reach out to me and we can potentially get you set up for giving a presentation during our session. Otherwise, abstracts are due January 31st, so you guys have got about 2 weeks to get those in if you're joining that party. And with that, task updates, we'll give an update on task progress, and then I'll pause briefly after each update for questions and comments. If you have a question after we move past the task, feel free to type it in the chat, and I'll circle back after all the updates are complete. So with that, I'll hand it off to Morgan Bond for carrying capacity standards update.
Good morning, everyone. So just a quick overview of what the carrying capacity standards team is working on. We're really trying to put together a document, a review document that is bringing together all of the different methodologies that people are using to estimate stream carrying capacity for salmonids in particular, and try and bring those together into one document, because in the past it's appeared that there's a lot of both interest in this topic, but also a bit of confusion and some difficulties with comparing some of these things across either methods or locations. And there's a lot of interest in it, so we thought it would be great to put together something that gives a little more guidance and information, and really kind of pulls together the expertise that we have across this region. So we have a number of authors outside of our team that are putting together sections for this. The document is in draft form now. It's quite long, and so we're really working extensively on paring this down into something that's a little more, I think, manageable and readable and ultimately publishable. So we've got all these sections that we pulled together. We're now working on putting together some tables and graphics. A really fancy graphic that you see on the slide here is a sort of a test graphic that we put together, that we actually have a graphic artist at the Northwest Fisheries Science Center that is going to be doing something a little more professional with this kind of thing. So we're putting together graphics and a few tables that I think will really help with the sort of readability and approachability for a larger audience for this. And then all of our individual section authors are going to be sort of helping to sort of pare down their sections and get this together. So we should have something, I think, later this spring or early summer that we will probably look for wider review of. And that's really where we're at. I'm happy to take questions if anyone has any questions or thoughts about this particular task.
Yeah, we'll just pause for a minute just in case you guys have any questions. And you guys can always reach out to the task leads if you have stuff you want to take offline, or hey Ralph, I see your hands up.
I had a question on the focus. Is this salmon focused, or does it apply to other species like lamprey?
Yeah, so the focus is typically, mostly the focus is towards salmonids, but the techniques that we're describing could definitely be applied outside of the sort of salmonid world for sure. And so I hope that it ends up being approachable for other species. Lamprey is a great example. Because of differences in life history, there would be some changes that people would need to make, but that's really true of any species. I think broadly, yeah, this should definitely be applicable to really any sort of stream dwelling fish. Yeah.
Awesome. Great question, Ralph.
Yeah, thanks.
Awesome. Well, we'll move on, and then if you guys think of anything, we'll swing back. Thanks, Morgan. And the next slide is the rotary screw trap task. We added a new component in 2023. StreamNet requested that the Fish Monitoring Work Group help gather regional biologists to demonstrate an Esri rotary screw trap dashboard that they're developing, and gather feedback on the tool. Nancy Leonard of PSMFC and StreamNet presented the dashboard to our group, PNAMP Steering Committee, and StreamNet Steering and Executive Committee. The Executive Committee has approved the tool with the consensus that the dashboard displays sensitive data discreetly and with agency understanding and approvals. Next, the StreamNet technical team will implement the recommendations, and the Fish Monitoring Work Group will reconvene the biologists to validate the accuracy of the data in the tool and just kind of verify that nothing is missing. Do you guys have any questions? And Nancy can help navigate too if you guys have specific questions. And then my team, just let me know if there's comments in the chat. All right, keep this moving along. Pit tag array data and related data analysis is focused on documenting and recommending improvements to data management and analytical methods and tools for pit tag array data. We coordinated with Pitagus to present this task and gather feedback on the needs of the regional community at their upcoming workshop. Marika Dovos with Idaho Fish and Game will give the presentation, and we'll work to identify issues and priorities from the attendees. And then the follow-up with the fish monitoring work group to kind of discuss some solutions, or if there's anything we can kind of do to help bios in the region get kind of what they need out of some of the tools, or maybe do some workshops or something. And there will be a link in the chat to the workshop. The workshop is this month, January 30th through February 1st, and registration is currently open but closes next week. So definitely sign up now. Questions on that task? And then that's the last task I'm going to talk about today. So if there's any questions or comments on the earlier stuff, just let me know. Alrighty. And with that, I am honored to introduce Ralph Lampman, the Yakama Nation Fisheries Lamprey project lead, to talk about all the cool work they're doing. The virtual floor is yours, Ralph. And let me stop sharing on my end.
Thank you, Megan. Let's see, let me start the presentation here. Let's see, does that look good? And should I keep my video camera on? Or
it's up to you. Yeah,
okay.
It's totally optional.
Okay. Yeah, I'll keep it on for now. So my name is Ralph Lampman. I work for Yakama Nation Fisheries, and today I'll be sharing updates about a lot of the collaborative and creative research projects that we're working on to help restore Pacific lamprey. I did pack in quite a bit, so hopefully I don't. Hopefully it goes smoothly. There's a lot of slides. Here we go. So just first, I want to introduce the team that we have. We have 5 full-time employees that work on our team, and they are we're a pretty small group, but we try to do as much as we can. All of them have lamprey attached to them. An outline. So I'll have a few slides, several slides for introduction, just to kind of get you acquainted. I want to introduce 7 projects that we're currently working on. I won't go too deeply into each one, but cover kind of the main take-home messages from the projects and where we're going next. And then we'll end with the kind of future needs and conclusions. And also Micah Pacific Lampman is my oldest son. He's also on my team, and hopefully he'll finish any project that I can't finish in my lifetime. The Pacific lamprey is a unique species, so they don't have any bones, they don't have any scales. They just have 2 dorsal fins, a caudal fin, no paired fins, no jaw, so they're part of the jawless fishes, similar to hagfish, and they just have 1 nostril on the top. They're really, ancient fish, so fossil records indicate they're likely 450 million years old or older. They're much older than dinosaurs. So, if we think about lamprey as a 100-year-old grandpa and grandma, basically humans are a 2-week-old baby. We're still in our diapers, although we act like we know everything on this earth. Pacific lamprey is not the only lamprey species in the region. There's also Western River lamprey and Western Brook lamprey. Even though they have a separate name, they're considered paired species. So they, similar to steelhead and a rainbow trout, they do mix and interact with each other. So they shouldn't be considered a separate species. They're kind of a species complex that they make up. The size looks the same on the top figure, but there's a big difference in their sizes. So Pacific lamprey is the largest one, and Western River lamprey is kind of in between, and then the Western Brook lamprey is only about the size of a pencil. It's pretty small, and the two larger ones spend some time in the ocean. There are many other species, especially in Oregon state. There are 10 species, so that's about 25% of the world lamprey diversity. This is a nice pamphlet by ODF and W introducing all these different species, and there's certainly a lot that we don't know about a lot of these endemic species. Pacific lamprey have three main life stages. So there's amosete, macrophthalmia, adult, or you could just call them larvae, juvenile, and adult. They do go through complete metamorphosis where they start off as the filter feeding or deposit feeding larvae in the fine sediment, and then they get their teeth and eyes. And then they head out to the ocean and then come back. So there's also what the tribes call night eel, which is likely referring to the fresh migrant adults that just came back from the ocean. And then they overwinter and they shrink during that time. And their colors also tend to change. And those are called day eel because their eye vision gets really weak, and oftentimes they'll show up like spawning during the daytime too. So they're not strictly seen during the night. They can come out during the day, so they're called day eel. Their lifespan is pretty long. So like compared to coho salmon, which has a 3-year life cycle, lamprey are much longer. I think we're thinking the latest research suggests that they're about maybe 10 to 20 years, maybe 14 as a kind of an average. They spend 3 to 9 years as a larvae, and then they overwinter for about a year. Or not overwinter. They transform into a juvenile, where they come out of the fine sediment, and then they start heading out to the ocean, and then they spend quite a long time in the ocean. Now we think the average is 5 to 6 years, and then they come back to the freshwater, often overwinter a year or sometimes 2 years or possibly 3 years before they spawn. This is the Hess et al. 2022 paper, and he has a nice figure showing kind of the lengths at these different life stages. So you can see, it's a quite wide range for larvae and juvenile adult stages, but adults are anywhere from 10 to 20 years old, maybe about, like I said, 14 years old on average. So yeah, but there's a lot of plasticity, and yeah, it could vary quite a bit depending on the river and the other features. So why do we need Pacific lamprey? For one, it's a really important tribal culture and food source. They've been harvested for millennium. But they're also an important food source for other species that are in the ecosystem. Lots of predators that are out there that feed on lamprey at different life stage. They are, they likely act as a buffer for salmon predation because the migration is often similar with anonymous submodids. So more lamprey could probably disguise the salmon that are migrating around the same time. Like you see in this example of the sea lions. And the, they're also, they serve a long time under the fine sediment, and they do quite a bit of work, even though we don't see much of it. They're, they help recycle nutrients that are they're detritivores, so they feed on things that are breaking down, and they help recycle those nutrients, make it available for bugs and other species that use that habitat. So just to showcase how important this is as a food source, this is a bald eagle snatching a lamprey out of the sea lion mouth. Yeah, that they're, if they don't really value the value lamprey as a food source, they probably wouldn't be doing that. They're also important, medical uses for lamprey. They have been used to, for anticoagulants to prevent blood clots. Spinal cord regeneration research is really dependent on the lamprey, and applying it to human research. There's liver diseases, high iron loading diseases, and even diabetes research. It's really useful, and just to understand how life began. It's one of the oldest vertebrate species we have on Earth, so it's a really important species, and there's lots of research that that sort of skyrocketed in more recent years for conservation and control in the Great Lakes, and then also for medical research. Just as an example of kind of the evolution, you could see that in the number of gills that lamprey have. So, most sharks have 5 gill slots. And this is believed to be because of the front 2 gill slots that that lamprey have. They eventually became the jaw for the fish, and that's why a lot of the sharks have 5, and lamprey still have all 7. So moving on to historical abundance, this is the Bonneville Dam estimated 24-hour count. So applied the night day time ratio to kind of historical numbers, and you'd see that the historical highs were close to 1 million. With Willamette River, there's probably another 1 million there that used to use that area, so likely 2 million or more in the Columbia River Basin. There was a really low period in the late 1990s to early 2000. The very low point was in 2009 when we had less than 20,000 total. So that's about 2.5% of the historical high. There's a petition for listing in 2003, and then there's this big black box of no data. About 30 years or so. There was probably a lot of lamprey to count, and Army Corps figured there's no point in counting these fish, so we don't know a lot about the data here. But what really helps us understand this period is the elder interviews. We have interviewed quite a few elders, and they provide really important information about how many lampreys are harvested locally in this period, and how it changed over time. Adult translocation began in 2000 by the Umatilla tribe and Pacific Lamprey Conservation Agreement. The first one was signed in 2012, so things really started to kick off around that time. There were definitely tribal harvest in the local area, and the Yakama Nation Treaty territories. There's the connection, that human connection, from elder to youth, and men harvesting the lamprey and women um, preparing them and cooking them, and a lot of knowledge and connection was there in these local areas. But there's also a lot of connection between the species, that are now lost in Upper Columbia. Many of the Upper Columbia watersheds, they were harvested in the Yakama River in several places until the late 70s to early 1980s, based on those interviews. And then they have become functionally extinct for over 30 years, at least in the Yakama Basin, until numbers have started to increase in more recent years. Those cultural knowledge is not completely lost. There's still people that know how to prepare the lamprey, and stories and legends are there. So we're hoping to hang on to those rely on those people that still have that knowledge, so we don't lose that. The 2022 assessment, this is kind of the latest map showing the rankings, the nature reserve rankings. You can see it's mostly orange and red, which are either critically imperiled or presumed extirpated. But with translocation, Okanagan and Similkameen did improve in the status. So from presumed extirpated to critically imperiled. Not certainly, there's a long ways to go to bring recovery, but hopefully we don't lose them in a lot of these local watersheds. But if we look further, in Canada and Idaho, they used to move much further upstream. So we probably lost about 50% of the historical distribution due to the existing dams. And at the uppermost dams, showcased by the red area, there's only about 100 adults that are making it to these dams. So they're functionally extinct in these places as well. These are some old photographs of lamprey from like Bruno River and Upper Snake River. When they dewatered the dam, hundreds of lamprey showed up there. And you can see in Kettle Falls in Upper Columbia, there used to be hundreds of lamprey migrating up, and based on these old photos. There are many threats that are that we have, like in Upper Columbia. These are the rankings that we provided. Main Stem Passage was kind of the highest. Climate change was second. But all these other threats are definitely there. Small population size, passage within the regional management unit, like in the tributaries, water quality, dewatering, habitat degradation, predation. Those things are certainly important. So starting with the collaborative project number 1, adult translocation is something that Yakama Nation has been working along with partners. In more recent years, the PUDs through the fish forums have started translocation, and the Colville tribe as well. So there's, with all the partnership, there's the numbers are increasing. In most recent years, there's been about 1,000 to 2,000 adults translocated each year. So these are the time period when other partners have began translocation. And the Hess et al. 2023 paper just came out. So this is a nice diagram kind of showing that we're now getting adults, or now we're now getting translocated adults, offspring, which survived the ocean, and they're now coming back to the Bonneville Dam. So we're able to detect, what percentage of them are coming back as adults, and also seeing, volitional adults, through our other monitoring, we're able to see where they're coming from. We still, over 75% of the pie graph, we don't know where they're coming from, but at least getting more information about their source and where they're coming from and where they're heading as adults. This table shows just kind of focusing on the translocation portion. So in 2019, we didn't have any adults returns getting detected, but 2020, we had our first few show up, and so these are extrapolated based on what adults we sample at Bonneville Dam. But now the numbers have really skyrocketed, and one thing that I'm proud to show is that Yakama Nation has 54% of the pie. So I think where we're putting the adults they seem to be really successful and coming back as adults. This is the changes in the distribution from adult translocation. So we did a separate analysis on distribution, where we don't incorporate translocation versus we do. So the purple areas, all the areas that have been occupied since the adult translocation began. So that's about, it's a little hard to see, so I'll zoom into each of these maps a little closely. For the Yakama, it's about the translocation began first here in the Yakama Basin, and in more recent years, we have also started early life stage outplanting using artificial propagation. But overall distribution have increased, have doubled or tripled based on these analysis, and the green and the red is showing where these adults have been released, and as adults in green, and early life stage outplanting as red. So those are mostly in the upper Yakama and the Cheese. And in the other watersheds in Upper Columbia, Wenatchee, Methow, Okanagan all have quite a bit expanded distribution due to translocation. So above Tumwater Dam in Wenatchee, in the upper reaches of Methow and Chewak River, and the entire Okanagan River. So again, distribution doubled or tripled in these subbasins. This is the number at Prosser Dam and also McNary Dam on the first McNary Dam on the first axis, Prosser Dam on the secondary y-axis. But after translocation began, numbers really have started to kick off. So we now have quite a bit more than we used to. So this is in the second lowermost rip dam on the Yakama River. But just to get a reality check on translocation, even though numbers have increased, this is showing the number of adults that are turned around at each dam. So Bonneville, the Dalles, and John Day. So about half of the population are turned around at Bonneville. So really, if we're really trying to ensure that these fish get to where they originally planned to, we would need to translocate 50 to 140 or 350,000 adults each year, if we're really trying to ensure these fish are not turned around at each of the dams. So our actual translocation numbers, even though increasing, are pretty minor and small in scale. We'll introduce the artificial propagation real quick. So this is a newer project. It started in 2012 in the laboratory. We finished a master plan in 2018, and the release actually started in 2021. So we have release for 3 years now. We believe it's a tool in the toolbox. It's, may not be the answer for everything for restoration, but there's certainly, a lot of important information that was gained from salmon hatchery, learning about their life history and the threats that they face. And I think this would be important tool, a toolbox for studies and potentially supplementation as well. These are the basins where we do translocation, and then the larval are showing kind of the early life releases. So like I said, upper Yakama and Nachis is where we're focusing the initial early life stage outplanting. These are kind of examples of the numbers that were released like in the first year. So the early life stage, like fertilized eggs, we certainly have more available, but as they get older, it requires more space at the hatchery. So there's kind of a smaller number, like for the age 3 larvae, or even the 4 months old larvae. So yeah, different life stages are released to understand, what is most effective in maintaining the fish at the release sites. I do promise we're not releasing these type of genetically mutated lamprey. They are, from wild source, and we try to ensure there's a diversity in the genetics from the propagation process. Number 2 is the adult passage. So we have worked on quite a few adult passage projects on the main stem as well as the tributaries. You can see like the wetted wall on the left. This is a vertical wetted wall that lamprey climbed to bypass the fish ladders. We also use like a 4-inch flexible tube to get them through the dam. Also use these flow refuge. They're just traffic markers that we add to the wetted wall, and it really helps slow down the flow. Kind of gives them a place where they can rest and then keep going. But in the typical fish ladder, there's a lot of 90-degree corners that adult lamprey have a hard time passing, so you can often see them congregating in these kind of places because the water is too swift and no good place to suck on to. But lamprey are really amazing in that they can, climb surfaces that salmon can't. Just on the wetted, there's a vertical wall at Willamette Falls, and you can see they often kind of go use each other to kind of help each other. So we call it the buddy effect. And when you have a lot more adults, they can kind of use each other and pass at a higher efficiency. So you can see some of that in this video. So we're trying to figure out, creative ways that we can, get those 50% that are turned around at Bonneville Dam and other places. This looks like a starship from Star Wars, but we call it the portable floating surface collector. Maybe there's a better name for it, but it's essentially a floating collector with 2 ramps, and water splashes onto the surfaces, and lamprey climb up those ramps, and then they fall into a channel where they then move through the 4-inch tubing and get to a trap box. So we're trying to test this at the Dalles Dam this year. It was a PLCI funded project, and we're hoping to get it anchored and tied to close to the east ladder entrance. lot of fish are likely getting turned around at this fast water entrance. There's a lot of 90-degree corners and slots that make it really hard for lamprey to enter. So we're hoping, those ones that give up, they can climb into the structure and provide a passage through that. This is the historical photo from 1937 at Bonneville Dam. You can see, if you zoom in here, there's like, I think I counted over 50 adults just trying to climb up this wetted wall. So, when they're searching for different ways to pass structures, this is what lamprey do. They look for these kind of splashing water and ways that they can climb around. So we definitely need to take advantage of this feature. So, in the 20 years or so of passage improvement at Bonneville, it has only improved by 10% or so, but we really need to get it up to 95% or higher. And at this rate, it's going to take hundreds of years. So we really need to think outside the box and figure out how we can make that happen. These are some of my napkin drawings of kind of creative ideas to get passage for lamprey. The left one is there is a lamprey orifice usually on the bottom of some of these dams, but maybe having one at the top could also help lamprey. So it's a little slot there to allow lamprey to pass through there. The right one is at the entrance, kind of envisioning just a trap box, narrow trap box place where lamprey can enter, and then the tubes, 4-inch tubes, allow them to go to the trap box upstream or at the top where people can access. Some of the PUDs have these creative mechanical traps that are in the fish ladders. So they're this one is at Daedalus PUD operated Wells Dam. Grant PUD also has some of these at Priest Rapids, but they just kind of slide into the weir slot and they can be quite effective. I think 10 to 20% of the adults are trapped this way at Priest Rapids. So potentially, this is something that we could do in the lower Columbia dams as well to ensure we get as many of those lost fish. Collaborative project number 3, juvenile larval passage. We have worked quite a bit on acoustic telemetry, pit tagging, or irrigation diversion, ways to reduce entrainment into irrigation diversion. One project I want to highlight is the Army Corps project. So the tribes have been pushing hard to that we get the survival study started and completed in the next few years. It started in the Snake River, and this year the plan is to look at McNary and John Day Dam, and then hopefully soon after we can do a study at the Bonneville and the Dalles Dam, so we can cover kind of the entire reach, not in 1 year, but at least over in segments we can see where the problem areas are. We would really like to do a study in the Upper Columbia as well. It would be great to have a joint study with all collaboration from all the PUDs and partners. This is kind of a potential number of adults that could be released to get that kind of survival rates at each of the dam, counting on the upstream release some of the juveniles to grow down into the lower dam as well, based on the Snake River study. But one thing we're finding is that there's limited trapping at dams in the Upper Columbia. There's limited juvenile that are collected at screw traps. A lot of them are larvae. and runtime can really vary for juveniles. So it's quite challenging to do the study unless we have a way to collect more lamprey. So we have collaborated with the PNAMP to design a trap that helps collect lamprey but avoids the collection of salmonids. This is kind of our initial design that we came up with. Jim Simonson did the initial napkin drawing, and he's a retired NOAA staff that has done quite a few adult passage structure designing, and really great partner. But so it's basically a bar screen. We can change the angle to kind of see what works best for salmon and juveniles, and change the spacing as well. And the idea is to test it in a flume and at PNNL, and then eventually after those testing, incorporate it into these specific areas within the dam environment, like the bypass flume or the raceways or the separator, underneath the separator. There's a few different places where they can go. This is my napkin drawing. You can see that I love napkin drawings. But potentially, maybe there's a way to incorporate this design for in-river trap. This is like kind of like a rotary drum, irrigation screen, except for kind of hoping that the lamprey will fall in and other species and debris are just bypass the system. So yeah, just putting some ideas out there. We have been starting to collect a lot of lamprey in the Yakama basins after translocation. So at Tanham Creek, Sadis, the numbers are still fairly low, but at lower Tompkins Creek, we're getting thousands of lamprey. And this is the 2021 results, but in 2022 we got over 4,500 lamprey in one day. It was just packed. The whole ski drop was packed with lamprey. So we're really glad to see that we're getting these kind of numbers, and hopefully in the upper Columbia there'll be more juveniles that could be trapped. Chandler also has a large number, so over 8,000 were collected in 2021. We do trap efficiency test. So this is showing top pitch and screw Sadis screw trap. We did some releases, limited number of releases. The recapture rates, there wasn't enough for Tanham Creek, but trap efficiency generally ranges from 0 to 16%, but more typically 1 to 6%. Lower Tompkins Creek being pretty high. So if we catch 3,000 lamprey and a trap efficiency of 10%, how many is migrating? Simple math tells us there's 30,000 juveniles that are passing the system. I think this is where we need to at least start and just get course data points, and I think over the years we'll have a lot more data to really understand the abundance of juveniles, but really encourage others to do the pit tagging and recapture so we can understand more about the overall abundance. So this is results from our pit tagging. It's a busy table, but mainly just showing a lot of these lamprey that we tag and release, they do get detected at Prosser Dam. Some are detected at McNary and John Day Dam. Some are detected at Avian Predation monitoring station. So they are definitely, it's not a big number, but if we tag enough, we do definitely get to see them. And we do collect genetics from the adults and the juveniles that we collect. So So these are kind of summary of how many genetic clips we took from this year, 2021. These are kind of examples of where the juveniles come from based on the parentage genetics. So you can see, even though Toppenish collects a lot, actually Sadis had a bit more on. So even though the screw taps are likely kind of low in efficiency, and so we're not able to see as much, but they are definitely there. But all the streams that we translocate in there, we are getting detection from those sources. We can also learn about ages. So some of these are moving out as age 2, but a lot of them are age 4 or 5. So this, which is kind of younger compared to other streams like in the Snake River. So it's definitely a really great learning tool to learn about their life history. Now moving on to predation. So we have done a predation study to understand the level of threat that lamprey face. We have started with the lab study initially, and now we're doing a field monitoring. So, lamprey are a pretty popular prey item. They don't have any bones. They don't have any scales. They're kind of like a worm, and you can pack them into your gut. So and they're really high in lipids and calories. So but oftentimes, because they don't have bones, they don't show up in the gut. Like if you're doing a gut content analysis, oftentimes they're overlooked because they degrade quickly. And so we have done a study in collaboration with Kramer Fish Sciences. We did a lab study to look at the detection level based on digestion hours, and we're able to see that by the time it reaches 50 hours or so, it's gone. But up to like 42 hours, we're still able to detect the lamprey from the gut. This is a northern pike minnow study, and but if you look at the gut content, like just after 7 hours, it's really hard to tell that it's even a lamprey. This is a lamprey on the left with a notochord, but hard to see like any of the gill feature, anything to tell that it's a lamprey. So definitely the molecular methods really help us expand that detection time. Something that we found from the lab. Smallmouth bass was a little different in that there wasn't a nice kind of clear declining trend, and even at 72 hours we were able to detect some. So it's kind of acted a little differently than the northern pike minnow, but regardless, yeah, we were able to detect them at these different digestion hours. In the field, these are the predator species that we looked at on the lower left. So American shad, channel catfish, largemouth bass, northern pike minnow, smallmouth bass, walleye, yellow perch. Just a few samples for yellow perch, but you can see they're feeding on a wide variety of predators or prey items. Pacific lamprey is a little hard to see, but I kind of showcase that they do show up in a lot of these predators, especially walleye. It was quite high. This is a map kind of showing all the different locations where we collected, and lamprey do show up. It's the blue segment here, but they do show up in some of the Columbia Basin sites in the Yakama River. So this graph was showing kind of the prey in families, but we do have actual species for each of these, so it gets pretty complex with individual species and kind of the season, and all that will be in our report. But yeah, some of the things we had to do was kind of create the threshold baseline level, because some of the these are bird species that were showing up. You know, they could be eating on, you know, the feces of birds or something like that, but it could just be in the water, and just could be a contamination. So we did increase the threshold size of the metabar coding results to weed out some of those in here. So we did compare metabar coding on the top, qPCR, eDNA results on the bottom. Lamprey were detected in you know, these various species. Like I showed earlier, they were you know, the qPCR was comparable to metabar coding, but for some species like steelhead, there was a huge increase in detection when we're using qPCR versus metabar coding. So there definitely seems to be some benefits in using qPCR. And then we had number 5, eRNA, eDNA versus bioacid. So we have also working with Kramer Fish Sciences, we are testing eRNA, so RNA and sampled in the water to see if we can not only detect lamprey, but you know, specific life stage of lamprey, like adult lamprey versus larval lamprey. And then the other project we're working on is cDNA versus bioacid from water samples to kind of understand the traction that adults have in the Upper Columbia Dam. So for eRNA study take home, we are learning that yes, you can detect specific lamprey RNA from water samples, at least in a lab setting. Spata 22_1 was the best candidate gene expression for detection of male adult lamprey. This is zoomed into the Spata 22A, so you can see, you know, for the mature male, the detection was highest, and there's a little bit of detection from the fresh migrant adults that have both females and males. There's a little bit of detection from larvae for whatever reason, but yeah, it's much higher in the mature males. So it could be, you know, a way to kind of decipher whether there's spawning adults in the system. The half-time of the RNA is quite short, so 12 minutes was the estimate. Efficiency for each step of is quite low, meaning that you really have to be careful in how you collect the eRNA sample, or you know, you just might not get the detection that you're hoping to get. So definitely protocols need to be improved in the future. So we're hoping to, you know, also understand what genes will help understand larval lamprey detect signals, and refine those protocols and do a field test. And then the eDNA monitoring that I mentioned. So we did sample like at Bonneville Dam as a baseline to kind of see where we know there's lots of lamprey. You know, this is the detection that we got. eDNA copies per second, so kind of higher in the lower below the dam and a little less on above the dam. Could be related to the abundance below and above. At Rocky Reach Dam, that's where we had some detection, but you know, no detection at a couple of the sites, and it did seem like, you know, those sites that were closer to the thalweg had higher detection, so that might be an important thing when we're sampling at these dams. But overall, you know, so we detected no signals from Wells Dam, which was kind of our target dam, because we were looking at the reduced number at Wells Dam and why adults are not, they don't seem to be attracted to the Wells Dam. They're turning around quite a bit before getting to Wells Dam. So, and then also there's limited detection, you know, in these tributaries, you know, just like right by the Midtown confluence at the Columbia River. There's small detection, but everywhere else it's pretty limited. So the eDNA, the top graph is showing the eDNA copies per liter, and then the bottom is eDNA copies per second. So it's basically multiplying by the river volume, the quantity. So the Bonneville Dam has certainly the highest and the lowest number of eDNA copies from the tributaries here. But after translocation began, we now have some detection at Wells Dam. So it does seem like the translocation has changed this picture from 2018. And we also started bioacid sampling, collaborating with the Michigan State University. So we're hoping to see how eDNA compares with the bioacid, which is the pheromone coming from primarily from the larval lamprey. And now moving on to the contaminant study. This is the we have looked at the mercury levels in 2014, working with PNAMP and CRITFIC. So we're kind of going back to a lot of these sites that had high mercury content in larval lamprey, and also trying to identify the source of mercury, whether it's a natural source or industrial, etc. These are the results from the 2014. So on the left, you can see that upper left, these are all the concentration of methyl mercury. It was really high in Clickitat for some reason. And if you look at Clickitat on the lower left from mouth to upper basin, it increased quite a bit as you got to this is close to the Clickitat hatchery area, but it was high. There's definitely the upper right graph shows kind of the correlation with the sediment concentration. So when the concentration is high in the sediment, it's high in the lamprey as well. And there's the bottom right is looking at the lengths of larvae and the concentration. So you can see that as they get bigger, you don't get that higher concentration. So potentially maybe these ones with really high concentrations are dying out before they get any bigger. We don't know that, but hopefully with the new study we'll have more answers. We also looked at the upper right graph shows the adult concentration, and there was a lot more in the mature adults after they overwinter, which was really interesting and concerning as well. We also looked at the eggs for the females, and they're about 4 to 13% vertical transfer of mercury based on the small samples that we evaluated. More, just recently, the Lamprey consumption advisory was came into effect. So OHA, Oregon Health Authority, announced that the combination of PCBs and mercury are at a concerning level, and so it requires some limitation on consumption. And then Washington State Department of Health also followed suit the next year. The graph or the picture on the right shows kind of what the one serving is. So it's a pretty small, less than, about a 1/3 to half of a adult. And child serving is even less. This is sad because lamprey are really a healthy food source. It's these are showing kind of all the different vitamins and minerals and fatty acids that lamprey hold. Just eating one lamprey, you pretty much meet all your recommended daily intake. There's some things like on the right shows vitamin A, B12, omega-3 are really, really high. So it's almost considered a medicine or superfood. So yeah, hopefully our rivers will get cleaned up, so we don't have to be concerned about the serving size. I think instead of one apple a day, we could have one lamprey a day. Um, uh, but certainly we need to bring back the numbers and ensure that they're safe to eat. So project number 6 is outreach, regional coordination. I think we do, in addition to managing the fish, we need to manage the people, and it's really important to get the word out about their importance and how we need to manage them. So we have lots of outreach events and presentations and media articles that we help put out. There's other creative outreach methods that are out there. You can run a marathon with a lamprey, or come up with lamprey songs, or use lamprey as your safety blanket at a dentist appointment. Yeah, you could choose how you want to get the word out there, but there's also, more recently we started lamprey in the classroom, where we had schools that have these aquariums. We provide the lamprey. They stay there until they get ready to spawn, and so we now have several classrooms that have these lamprey, and the kids are helping take care of them and learning from them. So I think it's a great, these type of interactions are really helpful and important for people to understand lamprey, what they are, and hopefully have better understanding about them. So next steps. Sorry, kind of my concluding slide here, but I think we do want to look at abundance goal. What is abundance that we need to strive to? We have been doing assessments through PLCI, but we don't really have a goal of what we're shooting for, and I think that's an important thing that we come up with in the near future. There's standard metrics that we could use to measure this, so like number per river kilometer could be one thing. We need to think about what level would provide tribal harvests in treaty territories, especially in the Upper Columbia, Snake River tributaries, where numbers are really reduced. What numbers do we need that would allow harvest and have a sustainable population? But we also need the lamprey simply for the ecological services to the stream ecosystem. It's something we need to develop. I think it's time that we use the habitat intrinsic potential models that U.S. Forest Service has come up with. They are great and they work really well, and I think it's time that each of the regional management unit adopts these and see where we're short on current distribution. I think we also want to make sure Pacific lamprey has equal footing as ESA species. Oftentimes, we are prevented from implementing a lamprey restoration simply because few salmonids interact with the lamprey structure, and but if it has tremendous impact on the lamprey, I think we need to think about as a whole how these lamprey species are helping salmon, and the overall impact, not just interactions here and there. So hopefully we can put them on a equal footing with these other species, and most first and foremost, I think urgency is something that the tribes really want to emphasize, that we can study them to death, but we really need to act quickly to make sure we come up with meaningful fast progression on their recovery. I think with that, this is just showcasing, with assessment, we do come up with abundance categories. So, you can use the median values to kind of estimate how many are in different regional management units. So I calculated there were about 250,000 in the Columbia River Basin based on the assessment, and Oregon Washington Coast provided, about 50,000. This is the intrinsic potential model that Forest Service has come up with, and the Western Washington on the left side and Eastern Washington on the right. The red shows kind of the really high ranking areas, or the probability is a lot higher, and it's mostly the kind of low gradient streams that have the best habitat for Pacific lamprey. But it also incorporates temperature and other things. And then finally, so I think we tend to just keep our blinder for salmon, but there's all these other species that are out there, and Pacific lamprey is certainly one of them. We really need to make sure we're restoring all these other community species that are out there. So maybe instead of Endangered Species Act, we need a Endangered Habitat Community Act to make sure we have healthy ecosystems, including crayfish. And then want to emphasize the strong need for partnerships. All these projects that I shared, I probably didn't fully explain all the partners that were involved. There were many partners, and all of us bring strengths to the to those projects, and really encourage all of us to think hard on what we can do for lamprey, and carry our share of responsibilities for the restoration. So yeah, this is the last slide for questions, and I didn't check time, but hopefully there's still time for questions.
Oh yeah, no, we've got like about 15 minutes. That was great. So we were getting some questions in the chat while you were presenting, so this is great. I can just kind of kick a few of these off. Casey Justice asked, do lamprey spend much time in the estuary, and what life stages are most common in the estuary environment?
Yeah, larvae are they can't tolerate salt water. They do increase their tolerance as they get bigger, but it's mainly juveniles that have finished the transformation that are using the estuary water, and also probably adults as they're coming back. They may spend a little bit of time before they come back up. Yeah, so yeah, definitely estuary is a key area, and an area that we don't fully understand how lamprey use. I'm sure it's an important area because there's more, host species that are available for juvenile to start feeding on. They kind of incrementally increase their host fish sizes. So estuary provides a lot more diversity of those fish sizes, and important place for them to get started on feeding, and then go on to bigger fish that are out there.
All right. Pat Connolly asked, regarding the 50% or so lamprey that turn back at Bonneville Dam, is there information about where they go, basically where they stray, and their fate, so reproduction and mortality rate relative to those that pass?
Yeah, that's a great question. I have the lower Columbia is on the kind of in the middle on the bottom here, so you can see their estimates of spawning abundance is really low. It doesn't amount to anything close to that 50%. So I think basically we just don't know what is happening to those fish, whether they're even spawning or yeah, where they get to go. It's really, I think the name Lost Fish is fitting because we really don't know what they do and how they contribute.
Interesting. Casey Justice commented too. Recent research in the Puget Sound has documented severe acute impacts from toxic compounds in streams such as PAHs and 6-PPD on salmon physiology and mortality. What is known about toxic impacts on lamprey?
Yeah, I believe there is someone from USGS that is interested in starting to look at Pacific lamprey. I'm forgetting his name. I can look it up. But yeah, that's as far as we know. I think we're definitely interested to learn the impacts from those contaminants.
Yeah, totally. And I'll have Ralph reach out with that name when he pulls it from the back of his brain. Diane Barton actually posted a link to a paper that was published about microplastics in lamprey. So if you guys saw that, that's in there too, and I'll include that in the notes. Ben Clemens asked, "Hi Ralph, if you had an additional large sum of money for lamprey restoration work or research, what would you use it on?"
Um, uh, yeah, that's that's great. I think we do need to focus on the key threats that are impacting a large number of lamprey. Bonneville Dam has the most number of fish and the largest number of fish that are getting turned around. So I think I would spend a good chunk of that money on trying to figure out ways to reduce that 50%. Looking at creative solutions and ways we can turn that around.
Yeah, that's a it's a big question. Diane Barton also posted, is there a difference between the day and night eel in nutritional value?
Yeah, that's a good question. Tribes do eat both fresh migrant and overwintered ones. Some people say that the overwintered ones actually taste better. Maybe they shed some of the extra lipids and maybe not as oily. But yeah, nutritional value, how that changes, I don't have good information. I think we looked at some of the contaminants in them. But yeah, perhaps that's an important thing to start looking at.
And then she had kind of a follow-up question. Do tribal members prefer day or night eels for their overall flavor?
Overall, what do you think for the overall?
Yeah, like would you prefer a day eel
Oh.
over a night eel?
Yeah, it probably depends on the person. Yeah, they're definitely, harvested right when they're coming back from the ocean, when they're big and bulky, but also they could be eaten when they're after they overwinter. I can't say one way or another. It kind of depends on the preference.
Totally. All right, that is all the questions from the chat. So if you guys have anything, I know sometimes it's a pain to type a question into chat. So if you want to raise your hand or unmute, you guys feel free. We got a couple more minutes with Ralph. So, all right, Ben Clemens. Looks like this for Diane. Dave Close published a paper on this where he interviewed Sea Tour tribe members and recorded their feedback on day and night eels. Interestingly, the Confederated Tribes of the Siletz Indians have published interviews on this as well, using the same terms. So that looks like somewhere where they're documenting some tribal knowledge, which is pretty cool. I'm assuming that's your kiddo. Go ahead, Jen.
I was just looking at Diane's question about preference, but she also asked about nutritional values. That's kind of interesting. Has anyone had the opportunity to look at nutritional values between, recently migrated from the ocean fish, Ralph, versus ones that have overwintered or are near spawning?
Yeah, they have. There has been a study looking at nutrients available in fish migrant versus overwintered. It was a graduate student project from Central Washington University, and they there were differences, and I can't recall the punchline, but it did change over time. But yeah, as far as like human consumption, how it changes, yeah, I'm not aware of any studies.
Thanks, Ralph. And thanks for being willing to take like any question that anyone can imagine about this complicated thing. So thank you for being game for this.
Yeah, absolutely phenomenal presentation. So much covered, so much great info on what's going on with lampreys in the region. I'm gonna give it one more second in case you guys are straggling.
While we're waiting, I'll just show. This is my oldest son eating lamprey for the first time, and this is from the South Coast Lamprey Summit along the Kukio River. They cooked up some lamprey for us, and my son was just loving it. He had a cracker and lamprey, but he was more interested in the lamprey. Yeah, we buckled him up so he can't escape, and so he finishes this experiment. Yeah, he got the cracker back, but then he just goes back to the lamprey. So
What more could you want? A lamprey and a cracker.
Definitely got your genes, Ralph. Love a lampreys. All right, awesome. And Ralph provided his email address, and I'll do that in the follow-up email. So if you guys think of something later, you can always reach out. I do have a quick question, Ralph. The lamprey hotline, what's that for? Just kind of chatting with you?
yeah, it's just me, but yeah, anytime it's open 24 hours a day. Whenever there's a question.
Awesome. Okay, excellent. If you guys wake up at midnight, maybe wait until 7 before you call the lamprey hotline. All right, I'm going to take control back and just kind of wrap some of this stuff up. We got 5 minutes left. I typically wrap up with some general fish monitoring workgroup information reminders. You can access more information by visiting the project page on PNAMP.org. The project page contains links to the task pages where you can find task descriptions, documents, upcoming meetings, links to previous meetings, ways to contact the task leads. I'll be working on giving the project page a full refresh in the coming weeks, so look forward to that. If you'd like to be added to the distribution list, or you have some interesting work you'd like to present, or have a presentation recommendation, go ahead and send me an email. That's awesome. We're always open to some feedback from you guys. The group is available to help you, so reach out if there's something we can do for you. The recording of this meeting will be available next week. This group will convene again on April 18th from 10 to 11 a.m. To close out for today, I would love to give Ralph a big thank you for presenting, and to our core team, task leads, and you guys, our lovely participants. We appreciate you guys, and stay warm out there.